I did not print stuff out, but I just filled it up and stuff.
That's fine.
In the 8th year of my God. So
Okay. All right, thank you.
No, you leave that there, girl. Don't make me like G-tips right now. I don't even know. It's been a long day already.
These ones found this
Illegal.
illegal. That's basically what we're doing.
So just listing a bunch of that and like a.
Yeah, it's listing facts. Yeah, and then it should say they found this.
And what does that mean? Is that something?
Yeah, it's not really
Yeah, my paper is kind of like that, to be honest. Like, I'm trying to like incorporate information, but it's just
That, like, where's my week?
I at least try to make this story like So, you know, I'm focusing on this like exocytosis. Like involve like proteins. So like this step involves this and like this will interact with the other one. And this interaction part is something I like. But at the same time, I was told that it's like a big connection according to this. So like. Okay, I'll look into it. I did my best.
That's what I'm saying, it's just like.
Feel that way.
Yeah, I sent it.
Take that into consideration.
Yes. You don't have power for only, you don't have it. That's all. No. Oh, do it. What are you doing that? I imagine when you talk about it, like, it's on a Google now. Before the meeting. We looked at it. Sunday and Friday are our regular days, but then it tends to be Thursday. It was 4 days on the calendar. Back to the original. I don't know what that was. So 4 days of these. Yeah. And then pencils are back on it. It's not anymore, it's over. Literally. Because I have to do that. Who knows? Who knows to say that? Until 2 a.m. Oh, yeah. He's gonna be like, I just have a few things I wrote down. They have manuscripts. Set them on that. So it's gonna be like, everything doesn't. Oh man. There's some other things we did wrong. I don't think on my mother packet to talk about.
All right, good morning, everyone. Okay, so today we'll start by going through worksheet 7 on the last 3 lectures, 15, 16, and 17, and then we can continue with the next lecture. That's going to be on more pathogenesis and microbe diseases. So there's a sign-in sheet going around. Just make sure you sign in just so that you can get points for this worksheet. And yeah, you can go ahead and go through those. And if you guys need extra explanation or visuals, I'm happy to pull up the other lectures and we can go through things within the lectures as well. So. Alright, so let's just go through, and once again, I would like to have everybody participate at least one question, yeah. So, what's the significance of microbial fermentation in food preservation? Yeah. Yeah, what part of the microbial fermentation inhibits the spoilage of organisms? So it's actually the acid production during that process, yeah. Bacteriocins is another mechanism in which those inhibit the growth of other organisms, but acid production through microbial fermentation, remember those short chain fatty acids, those are what inhibits the actual spoilage organisms. And so, yeah, so it's important to distinguish that. But if I were to ask you, you know, two different types of inhibition of growth of microbes on food, it would be the acid production through microbial fermentation and bacteriocins, which are these, you know, antimicrobial chemicals that are produced by bacteria, and they help to make sure other organisms don't grow. So, all right, what are the two most common probiotic species that we discussed in class?
Lactobacillus and
Yep, lactobacillus and bifidobacterium. Those are the two that you should know. If you look on any probiotic bottle, it's going to have at least one of those, if not both. So describe the process of pickling vegetables and how is this different from fermentation of milk products?
dairy fermentation includes starter cultures, which are added for flavor, texture, and texture.
Yep, exactly. So the important thing here is that in pickling, it uses these naturally occurring lactic acid bacteria that are naturally occurring on the vegetables. And so those, they're residing on the vegetables. And so those are what essentially is being used for pickling. And so the difference between fermentation milk products, like you said, Gabby, was that no starter culture was needed. But so yeah, it's a natural occurrence on the vegetables. Okay, list and briefly describe the 5 intrinsic factors that influence the growth of microbes on food during spoilage. What are the intrinsic factors?
Water availability, pH, nutrients, biological barriers, antimicrobial chemicals.
Yep. All right, and then the 3 extrinsic factors. Yep, storage temperatures, oxygen levels, and humidity. Any questions on any of these so far? Food spoilage? Okay. What's the difference between pasteurization and sterilization? Yeah.
Pasteurization kills specific microorganism. Sterilization kills all microorganisms.
Yeah, absolutely. That's a good distinguishing factor. Pasteurization will kill specific microorganisms that are pathogenic. Sterilization will kill all organisms. And then the actual process, the difference is pasteurization is just heating a liquid for a specific period of time. And so there's different kinds of pasteurization that we talked about. I gave you a table of these ultra-high pasteurizations and others. But it's just, essentially all of them include heating a liquid for a specific period of time. And then sterilization is like actually trying to eliminate everything. So there are levels of sterilization. If it's being sterilized, it's being sterilized. If it's being pasteurized, it can be pasteurized for varying temperatures, varying times. So there's differences there. Does that make sense? So microorganisms can be classified according to their growth temperature optimum. Define each of these classifications. So we talked about them being mesophilic, thermophilic, psychrophilic. So what are the differences there? What do those mean? Yeah.
Mesophilic have the mid-range temperature optimum. Thermophilic and hyperthermophilic have high temperature optimums. And then psychrophilic.
Yeah, exactly. So which classification of microorganisms, which one of these is largely responsible for food spoilage and when you're refrigerating your food? Yeah.
Psychotolerant or psychotrophic?
Yeah, psychotolerant, psychotrophic. So basically those are psychrophilic organisms that can actually still thrive in a little bit, colder or warmer temperatures than those psychrophilic organisms themselves. So those are called psychotolerant or psychotrophic. All right. We talked about both bacteria and fungi being able to spoil food, and they thrive in different environments. So which environmental conditions do fungi thrive in that bacteria do not?
Low moisture.
Yep, fungi thrive in low acidic and low moisture environments. So acidity and low moisture. And bacteria, we talked about, have trouble in acidic environments. The common example we've been using was the acidity of the stomach. So it's meant to keep bacteria from being able to grow there. And then low moisture environments, we know bacteria like moisture and they grow better in moisture, but fungi are able to thrive in the opposite conditions. So, all right. So any questions so far? We're here. Okay. Define intoxication and infection and how do each relate to the onset of symptoms.
Intoxication is a disease that results from ingestion of food containing the form of microbial toxin, which takes typically 4 to 12 hours for the onset, whereas infection is resulting from the ingestion of pathogen contaminated food followed by the growth of pathogen in the host, which will probably take like a few days to up to 4 days.
Yep, exactly. So so intoxication, a key thing there is that you're ingesting the toxin. It doesn't necessarily mean that the actual pathogen has to live and replicate inside of you. It just has to give that, produce that toxin in your body, and that's pretty much enough. And that's why you end up having the time of onset's typically shorter, within like a few hours, because that toxin is being generated. Whereas in an infection, you actually need the pathogen to be able to grow inside of the host or inside of you. And because it requires that, it's going to require a little bit more time to actually, show symptoms that typically take about 1 to 2 days, so 24 to 48 hours. All right, what are the factors contributing to someone's risk for developing a foodborne illness? Yeah.
What's the type of microbe or toxin ingested, inoculum size, inadequate stomach acidity, and reduction or change in normal microbiota?
Yep, exactly. Yeah, multiple different things can contribute to a person's risk, including their age, whether they're immunocompromised, and everything else that Erica said. Okay. So list 3 agents associated with food intoxication that we talked about, and where is the source of contamination found? Is it producing an enterotoxin or a neurotoxin? And then list 1 or 2 foods that are associated with each. So what is the first one that we'll talk about that's associated with food intoxication?
The first one was Staphylococcus aureus, and then the source of contamination was the nasal passage in a lot of humans, and it has an enterotoxin, and it's found in a lot of left out foods or mishandled foods by humans.
Yeah, exactly. So Staph aureus, this is the one that we've been working with in the lab. It's that gram-negative little grape-like structures. They're really small. It's a cocci. And so the majority of it is commensal, but in some situations you can have it, being on your skin or in your nasal cavity, and it could be inoculated during food preparation. So the source of contamination would be, a skin passage. And the toxin is an enterotoxin. So definitely, improperly stored or canned foods or implicated foods. We also see it a lot in mayo, mayonnaise, eggs, dairy, frozen foods. So there's a range of implicated foods for these because it's technically you're inoculating it during your food preparation. So it can come from your skin and be basically transferred onto different types of foods. All right, very good. What is the second one that we talked about, or another one that we talked about? Yeah.
The Clostridium botulinum, which source of contamination of soil and water, the botulism neurotoxin, and implicated foods are mushrooms, slime, properly canned foods.
Yep. Yeah, so C. botulinum, it has these endospores and it's found in soil and dust and water. And so it's a neurotoxin. So that's going to give you a complete different set of symptoms than an enterotoxin would. And implicated foods, like you said, are mushrooms, salami. And the other big one that was talked about was honey. Honey is a good source for that, especially in infants. So honey is, if we eat honey and it has this toxin, our body is able to clear it, but infants cannot. They haven't properly developed their immune system yet to be able to recognize that, and so they will be affected. So that's why you don't give honey to children. And what is the third one that we talked about? Yeah.
Bacillus cereus, and it's an environmental contaminant, enterotoxin, and lives within grains.
Yeah. So Bacillus cereus, it's an environmental contaminant, which means that it can be found in the soil. It's a heat-stable enterotoxin, just like S. aureus, but it's usually found in grains and rice. It's one of the main ones that causes intoxication from rice. Any questions on those? Okay. What is a disease characteristic or symptom associated with an enterotoxin as well as a neurotoxin? Yes.
I've got illicit net secretion of ions in water resulting in water diarrhea and abdominal cramping.
That's for the enterotoxin. Yeah.
I just said prevalent disease.
Yeah, so an enterotoxin, if you think about it, it's going to hit your enteric nervous system, so that's going to be your intestinal tract. And so basically you're going to have these abdominal cramps, watery diarrhea. It's going to really affect your stomach and your digestive system. A neurotoxin is going to, hit neurons and nerves, and so that's going to cause, paralysis in one of the worst forms. It can also just cause nausea, vomiting, visual impairment, respiratory impairment. You're not able to breathe well because it's paralyzing those muscles in your airway and your throat. It can impair your mouth or throat function. So those are very big differences there. It's not to say neurotoxin can't cause diarrhea, but it's not necessarily due to this net ion and water loss like with the enterotoxins. Neurotoxins can also cause constipation on the other hand, so that just depends more on the individual and their symptom outlook. But they both target complete different nervous systems. Okay. All right, number 14, fill in the table associated with each of the following foodborne infections. So salmonellosis, what's this causative agent, the species that causes it?
It's gram-negative enterotoxin.
Yeah, technically, I just wanted salmonella. Like that's what I was looking for. Yeah, not the specific one. Just salmonellosis. The causative agent is salmonella species. There's different salmonella species, so you don't have to know the actual details. And then what implicated foods? Like how can you usually get salmonella?
Like poultry and like cross-contamination with
Exactly, yeah, inadequately cooked poultry and definitely cross-contamination when you're cooking. And then some symptoms.
Nausea, vomiting, abdominal
So yeah, exactly. You're basically gonna have food poisoning. It's gonna lead to gastroenteritis. So your basically your gastrointestinal tract is gonna be just trying to get rid of it, and so you're just gonna have all of those symptoms. It's gonna get rid of it, one way or another. All right, and then Campylobacter infection. What's the causative agent? For this one, I have the full species name for you guys. So the salmonellosis, we said it was Salmonella species, so there's different types of species. For this one, it was a specific species of Campylobacter. Which one was it? Yeah. Yep, Campylobacter jejuni. And then the implicated foods, I guess I'll just say this one, it's similar to salmonellosis, inadequately cooked poultry. All of these really can be transferred as well through untreated or unpasteurized milk. And so, that's definitely something that can be a source for all of them, but generally Campylobacter jejuni infection occurs from inadequately cooked poultry, and it's the #1 cause of what type of symptom, or I guess like pathology? What symptom would people have? Yeah. Stool, headaches, nausea, abdominal cramps. Yeah. So it's the #1 cause of bacterial diarrhea. But all those things can happen, yeah. All right. And so E. coli food poisoning, we talked about E. coli being one of those organisms that's ubiquitous and it's found in a lot of different contexts. It can be non-pathogenic, like the one that we work with in the lab. It's in our guts, so we can, plate it out, and it could be a source of fecal contamination. But there's other really pathogenic forms of E. coli that we talked about. So that's the causative agent of E. coli food poisoning, just different pathogenic E. coli strains. So we've talked about that. But some implicated foods, this one differs a little bit, and then the symptoms of these really pathogenic E. coli strains. What are the implicated foods and symptoms? Yeah.
Meats, dirty water in food, unpasteurized milk.
Yeah. Yeah. Sorry. Go ahead.
And then you can have bloody diarrhea, which can lead to hemolytic uremic syndrome.
Yeah. So bloody diarrhea and ground meats are big ones for those. All right. And then listeriosis. What's the causative agent? Yeah, okay. Listeria monocytogenes, yep. Implicated foods.
Dairy meats, meat spreads, pasteurized milk, soft cheese, and raw fish.
Yep, lots of time it's like deli, meats, pates and spreads, unpasteurized milks and dairy products. And then the symptoms can range for this one. We talked about it being a special bug, and so because it's an intracellular pathogen, and so we'll talk about that a little bit more in lecture today, but because it's an intracellular pathogen, it can get inside of the actual host cell and infect that way. Its symptoms can vary from being non-invasive, gastroenteritis, so just regular like food poisoning symptoms where it clears, or they can be a lot worse. So what are those invasive or much worse types of symptoms that can be associated with listeriosis?
About like leading to meningitis.
Yeah. So bacteremia or septicemia, so basically you become septic. You have a bacterial infection in your blood. And so, once it's systemic, it's gotten into the bloodstream, that means, you're septic, and that's very life-threatening. It can also transfer the blood-brain barrier and get into the brain and cause meningitis. You can have endocarditis, so basically it can also transfer through the blood into the heart and cause a heart infection, and it could be really deadly. So that was one of those that you should know. Any questions on those? Any questions about intoxication versus infection or anything like that?
Question on one thing in the previous page.
Sure.
It was about fungi and how they grow. Is that they grow readily in acidic or low?
So yeah, good question. So acidic means low pH. So I guess what I wrote was low acidic, but it's really just acidic. Yeah. So what I should have said was low pH or acidic.
And low moisture.
And low moisture environments. Yes. Remember, moisture, the way that we can calculate that is through that water availability. And so, make sure you guys know that chart of water availability is pure water has a water availability of 1. And so if I were to give you something that has a water availability of 0.7, then that would be something that maybe fungi can take advantage of, but not bacteria, because that's a low moisture environment. So be able to read that table accordingly. All right. So number 15, define innate immunity.
Something about non-virulence, anatomical, physical barriers, as well as chemical barriers that are non-specific to illnesses and they're present to you first.
Yeah, exactly. So innate immunity is the first line of defense that your immune system has against any pathogenic invader. It's the first line of defense. So it's really ancient. It's evolved really early on, and even invertebrates. And this consists of everything from your skin, so the anatomic or physical barrier, to chemical barriers like enzymes in your tears and things like that. It's non-specific cellular defense, and it's present at birth. I'm just repeating things so this can be heard by everybody, and also in the recording as well. And then adaptive immunity, I guess I'll take this one, is it's not as immediate, so it's slower to respond. It's a more recent adaptation that we've evolved, and this is specific, a specific cellular defense in that it reacts to specific structures called antigens, and so our body has to detect those things before a response can be launched, so it's slower to respond. There's so many different antigenic structures, and this will develop over time after birth. Okay, so 17, just list some passive barriers to infection. So what are some physical barriers and some chemical barriers? I just went over a couple of them, but you guys can go ahead and give me some more examples of physical and chemical barriers. Yeah.
Lifestyle tears, secretions, also walls.
Yep, those are chemical barriers, right? And then what about some physical barriers? I already said the skin. What are some other, yeah?
Respiratory tract, urinary tract, GI tract, mucosal epithelium.
Yeah, exactly. Every, microbiome that you have in your body, like your respiratory tract, your urogenital tract, your gastrointestinal tract. Specifically, we talked about the GALT, system and how that works, and the mucosal epithelium, the mucus layer especially, and just the epithelial barrier itself. Those are all physical barriers that protect from invading infections. So although it's inside of you, it's still a physical barrier. Great. For chemical barriers, we talked about those enzymes. Also, acidity in the stomach. The pH of the stomach is really low. antimicrobial peptides are chemical signals, and those are produced by microbes and by our actual, immune cells and epithelial cells, and so those are also a chemical barrier as well. All right. What is hematopoiesis, and where does this process begin in the body? Yeah.
It's the process of creating blood cells.
Yep, it's the blood cell production process, and it starts in the bone marrow. What type of cells do the common lymphoid progenitor give rise to, and then what types of cells do myeloid progenitors give rise to? This was just from the chart. Yeah.
B cells and T cells. Myeloid progenitors, we have mast cells, myeloblasts, fibroblasts.
Yeah, so there's a lot there. The common lymphoid progenitor gives rise to B cells and T cells. Those are part of the adaptive immune system, so that answers 22, but there are also some cells that are part of the innate immune system that can be produced by the common lymphoid progenitors, like ILCs, or innate lymphoid cells, and natural killer cells, or NK cells. So the common lymphoid progenitors give rise to B cells, T cells, ILCs, and NK cells. And the myeloid progenitors give rise to neutrophils, eosinophils, basophils, monocytes. Those are all just leukocytes. And then there's like macrophages, dendritic cells, and mast cells. All of those are just part of the innate immune system. So for 21, which cell types are part of the innate immune system? It's everything except for the B and T cells. And the ones that we talked about that are part of the adaptive immune system are the B and T cells. Does that make sense? Give us a second to take your notes. All right. So 23, what is phagocytosis, and which cells have phagocytic capabilities? Yeah.
It's the process of engulfing microbes, and macrophages, monocytes, neutrophils, and dendritic cells.
Yep, exactly. For those of you who didn't hear that, the phagocytosis is the process of an immune cell engulfing a microbe via the extrusion of pseudopods. So it attaches to the pathogen and then it pulls it in. And then once that pathogen is engulfed, it puts it into a phagosome. So basically just a structure within the phagocytic cell that just encapsulates it. And then it's killed by lysosomes. So the cells with phagocytic capabilities are macrophages and monocytes, neutrophils, and dendritic cells. But neutrophils are special because we said that they kill cells in a different way. That's a little bit different from the general phagocytosis that we talked about that macrophages do. So how do neutrophils trap and kill the pathogen?
They throw a neutrophil extracellular trap of nets, and once it's trapped, an antimicrobial compound is used to kill the microbe.
Exactly. Neutrophils literally throw these nets. They spew a lattice of DNA and they trap it. So neutrophil extracellular traps. So instead of that, that's I guess in contrast to the extrusion of pseudopods in the actual cell, like pulling it into itself. Instead, it's kind of just throwing a net over it and trapping it, and then producing chemicals to kill it. And so we talked about antigen-presenting cells briefly, or APCs, and those are basically our immune cells that will take an antigen on a microbe and present it to a cell of the adaptive immune system to do something with it. And so which two types of antigen-presenting cells did we talk about? So I think the two we talked about were macrophages and dendritic cells. Those were antigen-presenting cells. There's something else, did I say something else or write something else in my lecture notes? Did anybody else have? You said what? You had what I said? Okay. Yeah, so I just want to make sure that we're all on the same page here. But yeah, the two types of antigen-presenting cells are macrophages and dendritic cells. Macrophages, they like engulf a pathogen through phagocytosis, and then sometimes instead of immediately killing it, they're able to present that microbe to another cell. So they have that added benefit of being able to like engulf it and trap it and present it. And then dendritic cells comes from the word dendrite, which means to bridge two things. And so basically that's how you can remember that it's an antigen-presenting cell, because it can present the cell and be that bridge to that second cell, immune cell, that it's presenting it to. All right, and so sometimes we have immune cells that don't really have the capability to attack the actual pathogen, but they will kill the host cell instead. So which cells will do that? Natural killer cells. It's in the name, those will kill the host cell instead of the microbial pathogen. All right, any questions on those? What are the signs, or the 5 signs leading to an inflammatory response, or just inflammation? Yeah. Yep. Heat, increase in temperature at the site, edema, which is swelling, redness, pain, and just altered function. What are cytokines and what is their function?
That bind to a specific receptor in terms of the immune responses to that. The immune response.
Yeah. Cytokines, they're just messages that tell more cells what to do. They can be proteins or small molecules that will bind to a specific receptor, and they do some intracellular signaling because they're messages. They can act locally or systemically. They turn immune responses up or down. They're produced by immune cells, epithelial cells. They're just, they're really important, but also always remembering that having too much of a good thing, so like having too many cytokines can result in a cytokine storm, and then that could really result to like multiple organ failure and death. And that's one of the main causes of death in COVID, is when your body is trying to really attack and ends up attacking itself and killing itself instead. All right, any questions on any of these? No? All right, so we'll go ahead and we'll continue on with the lecture. We'll go through part of this and we'll continue this on Wednesday. But I want to kind of reiterate some concepts that we talked about in the last lecture on innate immunity and infection. And then I want to also just talk about some more aspects of pathogenesis and microbial disease. so we talked about microbes having specific patterns on them in their, in their cellular structure. And so we call these microbe-associated molecular structures or molecular patterns. And so we call those MAMPs, and they're sometimes referred to as PAMPs in the case of it being a pathogen. So pathogen-associated molecular pattern. Examples of these can be things on the cell walls of bacteria, like the LPS or lipopolysaccharides. Those are part of the gram-negative bacterial cell wall. Teichoic acids are part of gram-positive cells. There are sometimes certain parts of the DNA motif, so an unmethylated CpG motif, you don't have to know what that is, but it's essentially, an unmethylated motif on bacterial DNA, but not on mammalian DNA, so our mammalian cells can't recognize it. Double-stranded RNA, that's part of viruses. Mannins, part of yeast cell walls. And flagellins, bacterial proteins, or flagella. So these are all different MAMPs, but they all have one thing in common, and that's the most important thing you need to remember. It's that these are structures on bacterial cells that we recognize as foreign because we do not have any of these. We don't have cell walls, so we don't have these LPS and teichoic acids. This is, specific to bacteria and not us. We are not a virus, so we don't have double-stranded RNA. We are not yeast. We don't have these cell walls in yeast, these components of their cell walls, and we don't have flagella. We have feet. So these are things associated with microbes. And so because these are going to be foreign to us, they are going to be recognized by our own structures as antigens. And so this is just another quick little image, just reiterating, how certain bacteria will trigger our immune cells. So here we have bacterial cells. This is a macrophage. So here we see the macrophage causing that extrusion and that pseudopod, and then it engulfs it. And so besides engulfing the actual, microbe, the macrophage can then also start producing cytokines and chemokines. Chemokines are types of cytokines, but they have a chemotactic factor, but just kind of to introduce a new term. And so this will cause vasodilation and increased vascular permeability, which just means that now these immune cells are being recruited and told there's something going on, so they will all come up and they'll start moving through, and so there's going to be more permeability in this epithelium. And so these inflammatory cells will then migrate into the tissue, releasing inflammatory mediators that will cause pain and redness. And so this is more of an inflammatory type response. And so we'll talk a little bit about different types of pathogens and what types of infections they can cause. So we've talked about Staphylococcus aureus a lot. It's again the one that we've been looking at under the microscope in the lab. It's a commensal commonly found on the skin. And sometimes, it can be transferred, like we talked about with food poisoning. It can be transferred from our skin into our food. But it can also, cause infection if it gets into a deeper layer of the skin. So if you have a cut or a scrape, and you have, Staph aureus getting into it, that can cause a deeper bacterial infection, and that's something we call cellulitis. And so it presents with these inflammatory responses, like swelling, redness, tenderness. It's going to be warm to the touch. And so just a reminder, this is what it would look like in a gram stain. They're gram positive, so they're going to be purple. I may have said gram negative earlier, I may have misspoke. So they are gram positive, they're going to stain purple, and they're cockside, they're clustered in these grape-like structures. And so another one that that I want to talk about is hookworms. These are more so like parasites, intestinal parasites. And so these are introduced to humans and other animals through the skin as well, typically the feet. These can be transferred in a lot of, water settings. If there's hookworm and you're swimming, then that can get through the skin. And so this one is different than the Staph aureus infection, for instance, because the other one just remains in the skin. It's just a deep bacterial infection of the skin, which we call cellulitis. However, hookworms can get in through the skin, but they typically make their way to the intestines through the bloodstream and the lungs, and so they use these teeth that they have to attach to the intestinal wall, and they get their food or they suck the blood from capillaries. And so basically, in order to keep the blood flowing and to keep getting that blood from your body, they secrete these anticoagulants. So basically, coagulants help to coagulate blood or make them more clumpy, and so that way you don't bleed out. But they secrete these anticoagulants so that the blood can keep flowing. And then they also have these proteases. So anything that ends with ACE, remember, is an enzyme, and enzymes break things down. And so they break down the blood for digestion for themselves. And so this altogether will result in an inflammatory response. But these are obviously more dangerous than the staph infection because they can make their way to your organs. And so every 6 hours, they need to select a new feeding site so they can like move on to a different organ and just keep sucking away and causing more damage. Questions? Ebola hemorrhagic fever. Ebola is not a bacteria, it's a virus. It's very contagious, and it's associated with hemorrhagic fever because it causes extensive hemorrhaging. Hemorrhaging is bleeding out, essentially. And so basically the way this works is it infects a number of tissues, including epithelial cells, but it does the most damage by infecting monocytes and macrophages. So this is one of those that can infect your actual immune cells that are trying to fight it off. And so this will lead to a lot of, a really robust expression of inflammatory mediators. So these are all associated with inflammatory types of responses, but you can see how they vary very differently. So this one, also results in the depletion of coagulation factors. And so you'll have really low blood pressure, cell death, which is the underlying cause of this hemorrhagic fever and this like bleeding or hemorrhaging. Does that make sense? So we talked about MAMPs and how they are specific structures on microbes that our hosts or us we can recognize. And the way that we recognize them is through these pattern recognition receptors, or PRRs. And so these basically identify the presence of the microorganism. So these are expressed on our, phagocytes and other cells in the body, and they include a number of types of receptors. So these are all receptors. They can be toll-like receptors. They can be just on the outside of the cell. They can be nod-like receptors. Those are inside the cell. There's some C-type leptin receptors and others. We're not going to go into details there, but there are a number of types of receptors, both extracellularly and intracellularly, that can recognize these MAMPs, or just the presence of microorganisms. But kind of turning this back over on the pathogen side, the pathogen will have specific mechanisms to do a variety of things. They have specific mechanisms to gain access and entry into the body and actually colonize and attain that unique niche for itself. They have specific mechanisms to acquire the necessary nutrients for themselves. They can multiply or persist through other mechanisms. They also have mechanisms to avoid, subvert, or circumvent the innate host defense. They can evade the acquired specific immune defense responses, so those are the adaptive immune responses. They can evade them. They can cause their own tissue damage or disease, and they can exit and transmit infection to new hosts. So these are all, or at least like the last 4 or 5 kind of are essentially mechanisms of virulence. And so mechanisms of these virulence are generally encoded by genes, so virulence genes, and having these genes distinguishes pathogens from non-pathogens. So these genes basically will encode for specific proteins that have specific functions that lead to specific mechanisms in which they can do these things. So we'll talk a little bit about the first one, that's infection that's initiated by gaining access to the body and colonizing, and that is usually done through adherence. So the pathogen has to adhere specifically to epithelial cells, for instance, so it has to interact with the molecules on the surface of itself and the host cell. And many different things can facilitate adherence. So, flagella, Peelle, fimbria, capsules, other cell surface adhesins. Sometimes they can produce, something in that environment that gives it an adhesin or quality. Just some pictures or examples of things. This is a capsule. You can see here this bacteria has a capsule around it. Flagella causes movement, so they can use this to attach. Peelle and fimbria are just other hair-like structures on the bacterial cell that they can use for adherence. So this is just a table giving examples of just specific attachments of different bacteria to cells or tissue surfaces. The thing that you guys should really understand is, you don't have to memorize these, but just the important concept here is that bacteria adheres through different things on their structures, and these adhesins have to attach to a host receptor. These attachments can occur at different sites, and they will lead to different diseases. Questions here? So biofilms are really important in infections because adherence and colonization of a microbe is mediated sometimes by biofilm formation. We talked about biofilms previously, so as a quick reminder, they are, when we have different communities of bacteria that are able to clump together and form, just a larger community of bacteria. And so they play an important role in chronic infections because they enable the bacteria to persist and adhere and resist other, defenses and antimicrobial agents. So biofilms are important in mediating this adherence and colonization. So pathogens have specific mechanisms to acquire nutrients and multiply and persist. So they need nutrients in order to be able to live and grow and eat and multiply and persist. And so in order to colonize, the pathogen has to multiply. And so the pathogen can grow either locally at the site of infection, or it can spread throughout the body, through the blood. Lymph nodes are usually a big part of the lymphatic system, in which your tissues and organs are connected to your bloodstream and other organs. And so a little bit of a tangent, but that's essentially how cancers metastasize. It's when they hit the lymph nodes. Once they get into the lymph nodes, they then have access to the remainder of the organs in the body because they've passed that barrier. But pathogens can do the same thing. They can grow locally at the site of infection, or they can spread throughout the body. And so the availability of the appropriate nutrients is the most important parameter that will affect the pathogen being able to grow. Nutrients can be limiting in vivo. Iron is a really important one. So our immune system has put into play this mechanism of iron sequestration. So basically what that means is a recognized innate immune mechanism that is against invading pathogens mediated by these iron-binding proteins called transferins. So usually the pathogens are trying to take up this iron, and so our immune system has these things in play, these mechanisms in play that will try to sequester the iron and keep it for itself. And so iron is a growth-limiting micronutrient for pathogens. The host has these proteins that bind iron called transferins or lactoferrins, lipocalins. Transferins is the one you should know. And these are very high affinity iron-binding molecules that function to sequester the iron in the host to limit infection, because these pathogens will thrive off of the iron. So we have these things in our immune system to prevent pathogens from taking the iron from us. But many pathogens then can produce iron chelating compounds, or we call these siderophores, to counter this iron sequestration. So, you know, we have this nutrient that can be limiting to pathogens, but they have a way around it through production of these siderophores. So here you can see this is a bacterial cell. These are our immune cells' way of producing these proteins that can then bind iron. And so, you know, bacteria can't really take this up, but it can then produce something. So right now, like the iron here is in red and it's trapped in these things that our body produces in these proteins, so we're sequestering it. And so here you can see the bacteria is producing this siderophore that can then break this down and pull the iron out and then take it up. Does that make sense? Any questions on this? So pathogens have mechanisms to avoid, subvert, or circumvent innate host defenses. And so we talked about capsules and biofilms. These can also help protect the pathogen against phagocytosis. So for example, Streptococcus pneumoniae protects the cells from being destroyed by phagocytes by having a capsule. So it's one of the examples that you should know. It'll form this capsule that won't allow it to be recognized by the phagocyte, because the phagocyte, remember, has to recognize something like a MAMP, or a microbe-associated molecular pattern, on the actual bacterial cell. And so having the capsule kind of hides that pattern out, so that pattern recognition receptor on the host cell can't recognize it. So basically there are these virulence or effector proteins that can directly be injected by bacteria into host cells. So what I'm showing you here is, this is a bacteria. So this is a gram-negative bacterial cell, so it has an inner and outer membrane. And this is a protein, for instance. And so one virulence factor, or one mechanism of making the pathogen more virulent and actually getting into the host cell, is by producing these proteins, for example, a toxin. And these bacteria have something called an injectosome. This is a type 3 secretion system, so it's a protein complex. I'll talk about that. But in gram-negative bacteria, this injectosome traverses both the cytoplasmic and the outer membrane of gram-negative bacteria. And so basically, these bacteria are able to inject this protein into the host cell through this injectosome. And so, you know, these virulence proteins or effector proteins, it just means that it has an effect on the host cell. And so this type 3 secretion system, or T3SS, is basically a molecular syringe. So these bacteria use a molecular syringe to inject proteins from the bacterial cytoplasm into the host cell. So here it's in the bacterial cytoplasm, and then now the protein is inside the host cell. This is a type 3 secretion system, and so it's an injectosome. So this is the mechanism of delivery. And so it actually appears that flagellar genes were evolutionarily re-engineered to encode proteins that act more like molecular syringes. This is just kind of a fun fact. Flagella are the structures outside of the bacteria, and so it seems like there's been some evolutionary adaptation in which you know, the cell has been able to evolve these type 3 secretion systems that will act more as like a molecular syringe. These genes are usually located on pathogenicity islands, so that's just essentially a part of the gene, a part of the chromosome. If you have like genes that are responsible for this type 3 secretion system, they're going to be found next to each other, and we call that like a pathogenicity island. These systems are found in a variety of different infections, and namely a few of the ones that we've been talking about, like Salmonella, E. coli infections, Shigella, Pseudomonas, Yersinia as well. We'll talk about Yersinia pestis a little bit. Yersinia pestis is the bacteria that caused the Black Plague. And so the reason why it was able to spread so fast and cause death in so many people is because it was utilizing this type 3 secretion system to block phagocytosis by our immune cells. So some pathogens can stop phagocytosis by injecting the phagocytic cell with effectors. And so Yersinia pestis does this basically by injecting that protein or effector, it's YAP, and that will prevent actin polymerization and blocks phagocytosis. So what does that mean? So over here we have Yersinia, which is the bacterial cell. And so these are just, different receptors on it that it can bind. And so this is the type 3 secretion system. So this is the syringe that allows it to basically get into the host cell, which is right here. So this is a macrophage. And like we said, macrophages usually will recognize something on the bacterial cell and that'll form these pseudopods and try to engulf it. But through this type 3 secretion system, Yersinia can inject these YAP proteins that block cytokine production and cell survival in the macrophage, but more importantly, they are able to disrupt cytoskeletal elements. So they disrupt the polymerization of the macrophage. So it starts to break down the actual ability of the macrophage to form these pseudopods and then engulf the protein. So that's how this works. Does that make sense? All right, so pathogens can also evade acquired immune responses, or in other words, adaptive immune responses, because they're acquired, they're not innate. And so we have antibodies that our body produces against pathogens, and that's all part of the adaptive immune system. We may or may not talk a little bit more about that. But some pathogens have ways to evade the host cell by actually hiding out inside of the host cell and remaining intracellular after being ingested. So antibodies generally within the adaptive immune response act on extracellular structures. So pathogens can evade that by getting themselves into the cell and remaining intracellular. So can anyone remember which intracellular pathogen I alluded to earlier that can do this and it becomes really deadly? Listeria, yep, exactly. Listeria monocytogenes. It's the cause of listeriosis. A little recap, it can lead to bacteremia and meningitis. It's found in soil and water, so in the environment. It's cycrotolerant, which means that it can grow at refrigeration temperatures. Usually it's killed by cooking and pasteurization. It's transmitted to people by food. The infection, once again, is called listeriosis, and it can cause severe and even mortality and death in vulnerable groups. And it's an intracellular pathogen. So basically, the pathogen is being taken up by phagocytes, like macrophages, and then it will be able to take advantage of that and grow and proliferate inside of those things. And so that's what makes its mortality rate so high, up to 20%. Less common, but it's a lot more deadly. And so, some pathogens that hide out inside of the host cell, they do this, and they are able to hide out by stopping the actual phagolysosome from maturing, or they can just escape the phagosome before it fuses with a lysosome. So this is a little recap of phagocytosis. And so here we have the phagosome. So this is the phagocytic cell. It can be a macrophage. And this is the bacteria. And first, the bacteria will bind to the surface through this extrusion of the pseudopods. So it'll extend itself and engulf the organism and ingest it. It'll eventually close off on it, forming a phagosome. So it invaginates and traps it within the phagosome. There's lysosomes as well in the environment that have enzymes, so lysozymes, and so those lysozymes will get into the phagosome, making a phagolysosome. And so then those things can then generate these reactive oxygen species and destroy the organism and then let it out and release the microbial debris. And so some pathogens will stop this phagolysosome from maturing, so they'll cut things off at this step, and some of them will escape this phagosome before it even can fuse the lysosome. So these are the two steps at which, they can escape, either escape or hide out inside of the cell. All right, so causing tissue damage or disease, we talked about certain virulence factors of pathogens. So they can basically promote infection and host cell damage. So just like we have enzymes that can, like lysozyme and things like that, that can break down bacterial cell walls and try to kill the bacterial cell, bacteria also have some enzymes that can break down our host tissue. These include proteases, nucleases, lipases. So proteases will break down proteins, nucleases will break down nucleic acids, lipases, probably lipids and other polysaccharides. And the very, common one we've talked about is toxins. So toxins are virulence factors, and so they can be exo or endotoxins. And this is what I was talking about on a pathogenicity island. So this is the plasmid DNA or the chromosome. You can have, virulence factors, and usually they're going to be kind of on the same chromosome because they're doing the same thing. So toxins kill or inhibit host cells. So endotoxins are usually lipopolysaccharides. Remember, LPS is found in gram-negative bacteria in the outer membrane. And these toxins can be released as bacteria are dying as kind of like a mechanism to try to survival mechanism. These can also serve as MAMPs, and they can bind to certain pattern recognition receptors on leukocytes or immune cells. And so once they are recognized by MAMPs, they can cause cytokine release. And so remember, a massive cytokine release can trigger fever, shock, and death. Exotoxins are also proteins that are toxic, and they're exotoxins, so they're released from the pathogen as it grows to cause host tissue damage. So instead of it just being released as it's dying, these are actually as it's growing, and it basically will poison the cell. And so it acts on host cells by a variety of different mechanisms. And so they basically will try to destroy the host cell by disrupting regular cellular metabolism. So endotoxins are disrupting through triggering these immune cells to make cytokines, and eventually you get too many, and then that can trigger, fever, shock, and death. So it's kind of endo or intra in a way that it's actually it's indirectly affecting your immune cell response. And exotoxins are a little bit different in that they are kind of causing the damage themselves by disrupting actual cell metabolism or destroying the immune cell. And then this is the last slide I have. Microbial exotoxins can be classified according to their mechanism of action. So they can disrupt the plasma membrane of the immune cell, the cytoskeleton, protein synthesis, cell cycle, signal transduction pathways, cell-to-cell adherence, vesicle traffic, exocytosis, superantigens, just a bunch of different examples, but they're all kind of stuff that the cell will need to do, and they will disrupt those mechanisms. All right, any questions at all on any of this? All right, I will see you guys on Wednesday. If you haven't signed in, please do so.